Hello students, myself Popere KS, Assistant Professor in Botany at Ray Shikshan Sanstha's RBNB College, Sri Rampur. In this lecture, we are going to start with the next topic, that is the cell wall. So, in the previous chapter, we have discussed about the cytoplasmic matrix constituent. So, first thing which we are going to discuss about the cell organelles or the cytoplasmic constituent is that the cell wall. So, now let us first start with cell wall. So, this cell wall it is the outermost membrane or the outer membrane of a cell. Now, in this cell wall, it is present in case of prokaryotes, so, cell wall, it is present in case of prokaryotes, fungi and eukaryote. And in eukaryote, only in case of plant cell. So, in case of the eukaryotic cell, it will be present only in case of the plant cell. Now, this the prokaryotic cells, they have cell wall as a material is so peptidoglycan, it will form the cell wall composition. It is consist of peptidoglycan. In case of fungi, the cell wall, it is made up of chitin, and in case of plant cell, it is made up of cellulose. So, cellulose is the most abundant carbohydrate which is present on the living earth. So, it is the most important and also present in means large amount or in abundance on the earth. Now, this cell wall, it was first discovered by a scientist, that is Robert Hooke. So, it was first discovered by scientist Robert Hooke in 1965. He has observed the cell in Koch cell. In Koch cell means the cell, Koch cell, it is in dead cells which are seen in the bark. In case of the trees, when secondary growth takes place or increase in girth takes place, it will form a tissue or a cell, that is bark, and this bark is made up of this Koch cell. So, it was seen like a like this, he has seen, Robert Hooke, he has seen the structures. These are the Koch cells, and he has seen hexagonal structures, and this outer layer, it is thickened structure. So, this is the cell wall. So, Robert Hooke first, or he was the first to observe cell wall. Now, next point, what is cell wall? Now, in case of the plant cell, the outer membrane is cell wall. So, in case of the plant cell, suppose this is a plant cell. So, the outermost layer will be cell wall. And so, the outermost wall in case of the plant is cell wall. Now, next to that, there is presence of the next membrane. This is plasma membrane. Okay, so next to cell wall there is presence of plasma membrane. Now this cell wall, it is present only in case of the plant. In animal cell, this cell wall is absent and they have the presence of plasma membrane. Outer membrane in case of the animal will be plasma membrane. Now this cell wall, it is a dead structure. So it is a dead layer. So this cell wall, it is mostly made up of a dead cell and it is rigid. So the cell wall is rigid and tough and this is dead. So cell wall is dead. But this plasma membrane, this is the living membrane. If from the plant cell, this cell wall is removed, so the material or the thing which left behind is the protoplasm, or it can be called as a living protoplasm. So if cell wall is removed from the plant cell, the next thing or the material which remain is the protoplasm, or we also call it as a living protoplasm. Now next to that, now the composition of the cell wall. Now this cell wall, the cell wall, it is this is formed So this is the structure of the cell wall we are going to see. So this outermost layer, this is the matrix. So this is it is called as the middle lamella. So this structure is called as the middle lamella. It is present next to the primary cell wall. So we are going to see the ultrastructure. If we see only the part that is the cell wall outer structure, it will be look like this. So the outermost layer, it is middle lamella. Next to that, there is presence of primary cell wall. And next to that, this is secondary cell wall. Now the secondary cell wall, it is made up of three layers. That is, first one is S1, next is S2, and the third one is S3. Now they all are made up of a similar composition, but only difference is that the layers or the laying down of the layer, it is in different orientations. Now after that, there is presence of one more layer. It may be present in some cases, or it may present it is absent in certain cases. That is the tertiary cell wall. So the cell wall, it can be divided into three. That is the outermost layer. This is the middle lamella. Now this is linking the two different cells. So from this side, now there will be a different cell. So these are between the two primary cell wall, there is presence of the middle lamella, or it is a cementing agent between the two different cells. Next to that, there is presence of primary cell wall. Now this primary cell wall, it is it is a homopolymer, or it is a polysaccharide, that is a homopolymer, means made up of similar polymer of glucose. So it is an homopolymer of D-glucose. It has the presence of glycosidic bond between the monomer unit or the glucose. Next to that, this secondary cell wall, it is a heteropolysaccharide, and it is also made up of this homopolymer, it is made up of cellulose. Next to that, this heteropolymer, this is also made up of different, that is the cellulose, or it is made up of xylose, mannose, glucose, galactose. So this primary cell wall, it is a homopolymer, but secondary cell wall, it is a heteropolymer made up of different, that is xylose, mannose, galactose and glucose. Next to, so these are the secondary wall, these are heteropolysaccharide, that is S1, S2 and S3. They have the same composition, only the difference lies in the, that is the laying of the layers. The layer, they are laid in a different orientation. So some may be straight or vertical, diagonal, so only the position of the or laying down of the layer is different. Next to that, this tertiary cell wall, it is mostly absent in the cells. Some cells may have presence of the tertiary cell wall. Now next to that, there is also presence of some extra material, that is in the plant cell, mostly they are made up of cellulose. They may also have presence of hemicellulose. They also have presence of pectin. Now mostly the cell wall, it will be consisting of this three material, that is cellulose, hemicellulose and pectin. But in some cases there is also presence of lignin, and there is also presence of some glycoprotein. Now this lignin, it is mostly means it will be deposited in the cell when it will become dead, or it will form a thick layer around a cell wall. When the cell wall become dead or the cell become dead, there is deposition of the lignin to outside of the cell. So there is deposition of the lignin and its size increases. So this is how lignin deposition will take place. And glycoproteins, they will be forming the different functions. They are present on the cell wall and they will do the enzymatic functions. So this is about the structure and the chemical composition of a cell wall. Now we will see the functions of a cell wall. Prior to that, how this cell wall is formed in case of the plant cell. Now formation of synthesis. During the cytokinesis process, cytokinesis means the division of the cytoplasm or the two cells. So during this stage, during the cytokinesis, now this two cells, it will have its own nucleus. During that stage, there is a formation of a cell plate. So this cell plate, this is also called as Plast. So this cell plate it is also called as phragmoplast. It is made up of from the particles or the remaining particles of the ER and Golgi. So fragments of ER Golgi, this will form the phragmoplast. Now phragmoplast means the cell plate. So from this is how the cell wall will be synthesized, or from the cell wall there will be a synthesis of a cell wall. So from cell plate there will be the origin of a cell wall. So this cell wall, suppose this is a phragmoplast or a cell wall. This is a cell plate here. So what will be happening? From both the sides there is formation of a primary cell wall. From this also there will be formation of primary cell wall, and now rest there will be again the formation of secondary and tertiary cell wall, and this will form the cell wall. So the synthesis of the formation of the cell wall, it is from the cell plate or the phragmoplast during the cytokinesis stage. Now next, the functions of a cell wall. Now functions, firstly it is mostly give mechanical support to the cell wall, because it is a rigid layer and it is the outermost covering of a cell wall, so it will give mechanical support to the cell, and it also give shape to the cell. And as it gives shape to the cell, on the basis of the cell there are different plant cells which are distinguishable, that is like parenchymatous cell, collenchymatous cell, and so on. So on the basis of a cell we can also differentiate the different cell due to the presence of the cell wall. Now this cell wall, as they have deposition of lignin and also there is suberin is present, so these are the material which are also present in the cell wall. So what they will do, they will not allow the water to pass through it. So they will prevent the water flow or the flow of water. So it will check transpiration in case of the cell. Now there is also this cell wall, it has a function, or it is a site for different enzymes. So all the enzymatic reaction, it will be occurring in the cell wall. So there are different glycoprotein, that is the enzymes which are present on the cell wall. So they will do the different enzymatic functions. And this cell wall also, when the cell absorbs maximum amount of water and it will swell up, but due to presence of the cell wall, it will restrict or it will prevent the cell from bursting. So during the endoosmosis, or when the cell it absorbs water, so what will happen, the cell wall, the size of the cell will increase and it may burst, but due to this mechanical support or the presence of the cell wall, it will prevent the cell from bursting. So this is about the functions of a cell wall and about cell wall. Thank you.